On last class, we studied about this vitamin D. Today we'll continue on this one parathyroid hormone. So this vitamin D parathyroid hormone. So we are studying these hormones on the topic this calcium homeostasis. Since these hormones are involved in the regulating this calcium concentration in our body. So this parathyroid hormone. So parathyroid hormone it is secreted, synthesized and secreted by a parathyroid gland. And I hope that all of you are familiar with a parathyroid gland. So where it is situated. So it is presents. There are four parathyroid glands that is situated adjacent to the thyroid gland, and the structure, the size of parathyroid gland is like a rice granules. And this parathyroid gland it is responsible for a synthesis and secretions of a parathyroid hormone, so which main function is regulations of body calcium. So it is involved in a body calcium homeostasis. So most of the times what happened is during a thyroidectomy, so that is the removal of thyroid gland, this parathyroid gland may also remove mistakenly, and which may lead to the hypothyroidism. It is. So now move on to the structures. So this while talking about the structure of this parathyroid hormone, so it is peptide in nature. So peptide means that is composed of amino acids. So the this parathyroid hormone it is also called as Colip's hormone, and it is a linear polypeptide which is composed of 84 amino acids and having a molecular weight 9,500 Dalton. So that is 9.5 kilo Dalton. So this hormone, so it have these N-terminal sites. So the sequences from 34 amino acid sequences from a N-terminal site. So that is the active active form of the hormones. So even if we hydrolyze this parathyroid hormone into the two segments, so that is from N-terminal, if it is partially hydrolyzed into two segments, that is from 1 to 34 to 35, and the rest segments, then the first segment, so which contains this first to 34 amino acids, so this is still active. So for its activity of the parathyroid hormone, so either it will be active in presence of the full segments also, and even the after the partial hydrolysis also, if it contains the first to 35 amino acids on the single segment, then it is sufficient for to showing the activity of this hormone. So this is the structures of pre-pro hormones. So pre-pro parathyroidism. So during a synthesis, so these like other polypeptide hormones, this hormone is also synthesized in pre as a pre-pro hormone. So that is the inactive hormones, and during the synthesis it contains 150 amino acids, and its molecular weight is 13 kilodalton. So that is 13,000 dalton. And this other this protein synthesized, this hormone is also synthesized by a ribosomes. So ribosomes of a rough endoplasmic reticulum, so as a pre-pro hormone. So when this pre-pro hormone it enters to the smooth endoplasmic reticulum, it removes the this signal peptide, so it is composed of 25 amino acids, and then it converts to the pro hormone, and to form a this one active hormone, so which contains a 84 amino acid sequence that takes place in a Golgi apparatus. And this the removal of six hormones from this N terminal, so that is done by the enzyme called as a colipase B or furin, so which is present in the Golgi apparatus. So so you can see and here in the line also. So as pre-pro hormone, preproparathyroid hormone, which contains 150 amino acids, and it is synthesized in the uh, polysomes, so ribosomes of a rough endoplasmic reticulum. And after that, it entered to the smooth this endoplasmic reticulum, where this 25 amino acid sequence is removed, and it contains 90 amino acids. So in Golgi apparatus, so with the help of the clipase B or furin enzyme, so there is a hydrolysis takes place on the N terminal. So 6 amino acids are removed, and it form as a active this one parathyroid hormone. And in the form of this active parathyroid hormone, it is uh, it is released into the cytoplasm, and it is packed in the secretory vesicle. And it while we studied during a thyroid hormone, so thyroid hormone also synthesized in a same pattern, and it is also stored in a secondary vesicles. Unlike thyroid hormone, the storage of this parathyroid hormone is very small or very less. So while talking about the thyroid hormone, it can be stored for a longest period in a thyroid gland itself as the form of a secondary vesicles. When talking about this parathyroid hormone, it cannot stored for a long period and in a large amount. So it is stored in a small amounts and for a small period only. And this synthesis and secretions of this parathyroid hormone, it is regulated by a concentrations of the calcium. So low concentrations of a calcium stimulates the synthesis and secretions of parathyroid hormone from a parathyroid gland, whereas high concentrations of the calciums in a circulations, it inhibits the synthesis and secretion of this parathyroid hormone from a parathyroid gland. And this is also a contraindicatory point. So we'll discuss on a later. So this the secretions of parathyroid hormone from a parathyroid gland is regulated by extracellular calcium. So that means the calcium is present in a extracellular fluid. So that is in a presence in a plasma. So decrease, so low level of calciums in a plasma stimulates the secretion of parathyroid hormone from a parathyroid gland, and the high concentrations of calcium present in a serum inhibits the secretions of parathyroid hormone from a parathyroid gland. And the rate of these secretions of parathyroid hormone from a parathyroid gland, it is directly related with the concentrations of the calciums present in the serum or plasma. So this the secretions of parathyroid hormone from parathyroid gland is rapidly increased when the calcium concentrations fall below 9 milligram per deciliter. So the normal level of the calcium in a serum is 9 to 11 milligram per deciliter. So on that, generally what happen in serum, this calcium is present in a three form. So 50% of the total calcium that is present as a ionized form, and 40% of the calciums that is bound with albumin, and remaining 10% of the calciums present in a serum, they are bound with a another these molecules, that means in the phosphates or others as a, so bound with the other compounds. And the secretions of PTH is directly interfered by the ionized calciums, not by the this bound form of the calcium. So these PTH secretions, so it is increased rapidly on calcium level when it falls below 9 milligram per deciliter, and reaching a maximum at 6 milligram per deciliter. And if the calciums fall below the 6 milligram per deciliter, then it will not it will not affects more on the secretions of parathyroid hormones from a parathyroid gland. So, and this increased calcium's level depresses the hormone level. So it inhibits the secretions of parathyroid hormone from a parathyroid gland, and the maximum these inhibitory effects can be seen when the calcium's level at 11 milligram per deciliter. And however, the secretions is never completely suppressed. So it cannot be completely suppressed. Even the calcium is increased more than 11 mg per deciliter. So this is not completely suppressed. So regulations, as we discussed, the main compound which regulate the synthesis and secretions of parathyroid hormone is that is the calcium. So decreased level of in a circulations, it stimulates the synthesis and secretion of parathyroid hormone from a parathyroid gland, and this in physiological conditions, that is in a physiological conditions, this low amounts of this magnesium ion and increased phosphate concentrations in a circulations also stimulates its release from a parathyroid gland. And this parathyroid hormone release is stimulated by decrease in cytosolic calcium, which is contrast to most cell. So when you have studied these lots of these proteins, these peptide hormones, so like these thyroid hormones, so when they are stored in a thyroid gland, so for releasing, that means exocytosis of this thyroid hormone from a thyroid gland require a high concentration, that is the entry of calcium to the thyroid gland. And this release of parathyroid hormone is contrast to those activity. So this the release of parathyroid hormone from parathyroid gland is stimulated by a low concentrations of calcium, cytosolic calciums, so which is contrast to the most cell. So if there is the increased concentrations of calciums in a cytosol of this parathyroid gland or parathyroid cell, it stimulates the degradations of stored parathyroid hormone. So in case of other hormones like this thyroid hormones, so which is peptide in nature, so the releasing, so exocytosis of these hormones from a parathyroid gland, it is well, it takes place when there is the increased calcium concentrations in the cell, and this is the contrast of that function. So the release of parathyroid hormone from the secretory vesicles only possible when there is the decreased concentrations of calciums in the cytosol, so that is in a cell. So in human beings, this parathyroid hormone secretion is suppressed by a severe hypermagnesemia, even when accompanied by a hypocalcemia. So extremely low level of magnesium also severely decrease the secretions and peripheral effectiveness of parathyroid hormones. So apart from these calcium's level, magnesium level, and phosphate, it is also affected the secretions of this calcium. So the secretions of parathyroid hormone is also affected by the catecholamines, and generally these catecholamines they act through a beta adrenergic receptors, and these cyclic AMP levels have also a modest stimulatory effects on the parathyroid hormone secretions. So reduced cyclic AMP level, it prevents the release of these PTH from a secondary vesicles. So degradations of this parathyroid hormone. So these degradations mainly occur in liver and kidney. So generally this hormone is cleaved into the two segments. So one is N-terminal portion, so which contains the first 35 amino acids, which is still active, and the remaining molecules. And remaining molecules, they are inactive, and then it is excreted eliminated to the in the urine. So the half-life of full disparathyroid hormone and active segment, it is around 2 to 4 minutes, and they are completely degraded in the target tissues, kidney and bones, and completely degraded into the free amino acids. So the inactive segments, so it have a longer half-life, but it is a metabolically inert and is excreted in a urine. So that is eliminated via the urine. Mechanism of actions. While talking about the mechanism of actions, in the targeted cells of the body, so they generally contains two types of receptors for a parathyroid hormones. So this parathyroid hormone 1, parathyroid hormone receptor 1, and then parathyroid hormone receptor 2. And this parathyroid hormone receptor 1 is mostly predominant in this kidney and the bone. And this parathyroid hormone 1 receptor, it is also recognized by the another protein called as a parathyroid hormone related protein, PTHR protein. And uh, the metabolic effects of this parathyroid hormone related protein will be discussed on a later slide. And generally, these hormone, parathyroid hormone, they bind with the specific receptor presence on the target cell. So that is a this parathyroid hormone receptors. So when these parathyroid hormone, it binds to the receptors, these receptors, so generally the receptors of this parathyroid hormones is the uh, heptameric receptors, so which is coupled with the G protein. So when they are bind with this receptor, it activates the G protein. So either these two G proteins, one is this GQ proteins, and another one is that is the GS protein. So when it activates the GQ proteins, this GQ proteins, it stimulates the effector protein called as a phospholipase C, and this phospholipase C, it cleaves the phospholipids into the this diacyl glycerol and inositol triphosphate. And this diacyl glycerol and inositol triphosphate, which is the second messenger, they cause the increased cytosolic calciums and cause activations of protein kinase C. And where this protein kinase C, it causes the phosphorylations of the targeted enzymes. So apart from that, it also activates the GS proteins, and the GS protein, it activates the effector protein called as a adenyl cyclase, so which increases the concentrations of cyclic AMP. So cyclic AMP is the second messenger, which activates the protein kinase A. So inositol triphosphate and diacyl glycerol, so they increases the cytosolic calciums and also activates the protein kinase C. And this cyclic AMP, which is another second messenger, that activates the protein kinase A. So action. So the main functions of this parathyroid hormone is to contribute along with the orders to maintain the concentrations of calcium in extracellular compartment. So the functions of this parathyroid hormone is calcium homeostasis, and for that it targets bone, renal tubules, and also the vitamin D metabolism. So the main function of parathyroid hormone on the bone is its effect on bone is it, it mobilizes the calcium from bone. So that means bone resorptions. So to release calcium from the bone, it generally this parathyroid hormone, they are involved in generally in two processes. One is osteolysis. So osteolysis means that is the releasing of calcium from a bone, and another one is the osteoclast resorptions. So osteoclast means that is the degradations of bone. So generally in bone, two types of cells are there. One is osteoblastic cells and another one is the osteoclastic cells. So osteoblast, that is involved in the mineralizations of bone. So that is the formation of formations of bone and then depositions of minerals over the bone. And osteoclast, it generally causes the demineralizations of bone. So calcium's release, so which is called as a osteolysis. For this activity, parathyroid hormone, it stimulates the calcium ATPase enzymes and cause a active transport of calciums to the extracellular fluid. And the responses of these parathyroid hormones occur within a minutes. And for this activity, it also require a calcitriol. So this calcitriol is a active form of vitamin D. So the releasing of calcium from bone to the extracellular fluid, it require a activity of vitamin D also. So next function, that is the osteoclastic resorptions. For that, so there is parathyroid hormones, it causes the increase activity of glycolysis and TCA cycle. So citrate cycle. So which causes a increased productions of citrate and other organic acids. And when there is the acid acidic conditions over the bone, it favors the bone mineral resorptions. So apart from that, this parathyroid hormone, it also stimulates the differentiation of osteoclast precursor cells and activates the productions of cytokines, so which stimulates the bone resorptions. And the process of these osteoclast resorptions, it requires hours to day. So it's a slow process. So now effects on kidney. So this parathyroid hormone, it stimulates the calcium's reabsorptions in nephron distal convoluted tubules and decrease calcium excretion in urine. So in normal conditions, that means in while talking about these calcium's reabsorptions, normally what happens, 90% of these calciums, so they are reabsorbed in a proximal convoluted tubules. So actions is which action is not affected by parathyroid hormone. So once you can listen that. So the calcium reabsorptions. So majority of calcium reabsorptions in a kidney, that takes place in a proximal convoluted tubules, and the activity is not affected by the parathyroid hormone. And these absorptions of calcium, which is takes place in a these proximal tubules, it is absorbed along with the sodium. And the 10% of calciums is reabsorbed from a nephron distal convoluted tubules, and the absorptions is interfered by a parathyroid hormone. And this 10% of calcium which is reabsorbed from a distal convoluted tubules, so it affects the level of these calciums physiologically. So this the regulations of calcium's reabsorption by parathyroid hormone is necessary, or it is a beneficial. And apart from that, this parathyroid hormone, it reduces the phosphate reabsorption in a proximal tubules and to a lesser extent in a distal nephron. So apart from these demonizing that inhibitions the reabsorptions of phosphate in a proximal tubules. This parathyroid hormone, it also reduces the renal absorptions of potassium citrate and bicarbonates, so which generally produces a discrete acidosis. Apart from that, it also reduces the excretion of uric acid in a urine. And another thing what we have to remember is that phosphate excretion is essential. That So if phosphate excretions is not occurred by this parathyroid hormone, then what happen? This hypercalcemia and the hyperphosphatemia takes place at the same times, and which can produce a this one which can produce a this one soft stones, that is the calcium, which causes a this calcium phosphate precipitations. And these calcium phosphate precipitations, that is the source of stone formations in the soft tissues as well as in the renal tubules. And these excretions of phosphate during a hypercalcemia, it prevents the these formations of the stones. So these parathyroid hormones, it excretes the phosphates from a urine, while it prevents the excretions of calcium at the same time. So effects on vitamin D metabolisms. So generally what happens, these conversions of provitamins, that is a cholecalciferol, so generally we call as a this provitamin. So this vitamin D to the active form, that is the calcitriol, so that is the 125 dihydroxy cholecalciferol, so that is takes place in a kidney. And for that, it require activity of the enzyme called as a 1 alpha hydroxylase. And this parathyroid hormone, it stimulates the 1 alpha hydroxylase enzymes, so which converts the vitamin D to its active form called as a calcitriol. And this calcitriol is, it acts on the intestine, which increases the which increases the absorptions of calcium and phosphate from a intestinal lumen. And the source of this calcium and phosphate is generally from a dietary source. So from that, here this parathyroid hormone, it increases the absorptions of calcium and phosphate, but the activity is not direct. It is a its activity against its action on intestinal intestine is the secondary. So it absorbs calciums and phosphate from a dietary source in a small intestine, but actions of this parathyroid hormone is indirect, not a direct. So for that, it increases the conversions of vitamin D into the active form, and in turn, this active vitamin D, it enhance the absorptions of calcium and phosphate from a small intestine. So this parathyroid hormone related protein, so which is also called as a PTHRP. So this parathyroid hormone related protein, it is normally produced by fetal and adult tissues. And this the sequence and terminal sequence of this parathyroid hormone related protein is, it has a high homology with a parathyroid hormones, and they have a affinity to the receptors. So the affinity to the this parathyroid hormone receptors presents on the bone and kidney. So which we discussed on the earlier also, this parathyroid hormone receptor 1, which is called, so it have a this receptor 1, it have a affinity with a parathyroid hormone related protein along with a parathyroid hormone. So the another receptors, that is parathyroid hormone receptor 2, which have a only affinity with a parathyroid hormone, not with a parathyroid hormone related protein. And this parathyroid hormone related protein, it promotes hypercalcemia and decreased phosphate and cyclic AMP concentrations in kidney. So like these parathyroid hormones, this parathyroid hormone related protein, it also causes a hypercalcemia and hypophosphatemia, like done by the parathyroid hormone. So activity is similarly on the kidney. So in intrauterine development, this parathyroid hormone related protein, it regulates the chondrocytes proliferations and mineralizations and cause a calcium transport to the placenta, transport in placenta. Protein, so it it regulates the chondrocyte proliferations and mineralizations and calcium transport in placenta, so during a intrauterine development and after birth. So after birth, it contributes to the development and cell differentiations of this mammary gland, skin and pancreas. It also has cytokine type effects during inflammatory response, and which causes a this vaso smooth muscle relaxations, and it also acts as a neuroprotective during a aging. So under higher concentrations, it reduces the calcitriol formations, uncoupled bone resorptions, and new bone formations, resulting severe loss of bone tissue. So under normal concentrations, that under physiological concentrations, it involve in the adult, it is involved in the development of this mammary gland, skin and pancreas. But if it is present in a higher concentrations, it reduces the calcitriol formation. So that is the vitamin D active form of vitamin D formations, and so which lead to the uncoupling of bone resorptions and new bone formation, resulting the severe loss of bone tissue. So in normal physiologic conditions, it helps in the mineralizations of the bone, and in higher concentrations, it causes a bone resorption. So now these disorders of parathyroid hormones. So like in the other hormones disorders, so decrease concentrations of parathyroid hormones in a circulation, so that is called as a hypoparathyroidism, and increased concentrations of parathyroid hormone in a circulation is called as a hyperparathyroidism. So first, this one hypoparathyroidism. So the main cause for this hypoparathyroidism is that is the unintentional removal of the parathyroid gland during a thyroidectomy. So along with the thyroid gland, so there is the high chances of this removing of this parathyroid gland also. So since these parathyroid glands, so they are also attached with a thyroid gland. And when the persons have a hypoparathyroidism, it is characterized by a decreased serum calcium and increased serum phosphorus. So hypocalcemia and the hyperphosphatemia are the characteristics of this hypoparathyroidism. So there is a when there is less concentrations of parathyroid hormones, then this in that case it promotes the calcium phosphate deposition in bone, and it also uh, reduces the excretions of bicarbonate. In absence of this parathyroid hormone, there is the reduced excretions of bicarbonate and may lead to the alkalosis. So high concentrations of parathyroid hormone, it reduces the reabsorptions. So that means it increases the excretions of bicarbonate and generates the acidosis. But in deficiency, what happen? Decreases the excretions of bicarbonate and may lead to the alkalosis. So clinical symptoms of this hypoparathyroidism is that is the neuromuscular hyperexcitability, and it can be manifested by a paresthesia of fingers and toe, so cramps, so reduced myocardial contractility with a prolonged QT interval. So there will be the irritability, so tetany and convulsion can be seen in case of hypoparathyroidism. So pseudo hypoparathyroidism. So in this case, pseudo hypoparathyroidism, there is the when if we measure concentrations of parathyroid hormones in the circulations, we can found a normal or increased parathyroid hormone level. But due to the lack of the receptors on the target organs, these parathyroid hormones they cannot choose its effect on the target tissues. So which lead to the hypoparathyroidism. So that's why it is called as a pseudo hypoparathyroidism. Though the concentrations of hormone level is normal or increased, but it shows the symptom to the hypoparathyroidism. And this the cause for this pseudo hypoparathyroidism is that is the it is a rare disorder, so rare genetic disorder in which these parathyroid hormone receptors are affected, and so that due to the lack of these receptors, these have hormones have a resistance to the target tissues, so they cannot able to show the functions to the target tissues, so that lead to the hypoparathyroidism. So next, this one hyperparathyroidism. So hyperparathyroidism means that is the increased parathyroid hormones level in the circulations. And generally, this hyperparathyroidism can be primary or secondary to the other pathologies, including parathyroid functioning tumors. So when this there is the this parathyroid this adenoma takes place in a parathyroid, then it increases the synthesis of parathyroid hormones. So increases the synthesis and secretions of parathyroid hormones, so which lead to the high concentrations of parathyroid hormone in the circulations. And kidney failure or in a nutritional deficiency of calciums, so which also causes a when there is a nutritionally deficiency of calcium, then hypocalcemia, it causes the increased synthesis and secretions of parathyroid hormone, which lead to the hyperparathyroidism. So this hyperparathyroidism, it may also seen in case of the certain cancers like this one, these lung cancers and other cancers, where there is the regular where there is an increased synthesis of this parathyroid hormone related protein, and this parathyroid hormone related protein, so that also have a this one affects like a these parathyroid hormones in a target tissue, so which causes a which shows this different characteristic caused by the hyperparathyroidism. So that means this when the persons have a hyperparathyroidism, it shows a characterize of a increased plasma calcium level, that is the hypercalcemia, and also cause as a hypercalciuria and hyperphosphaturia. So when there is a this excess parathyroid hormone, then it causes a regular resorptions of a bone for a releasing of calcium from a bone, which lead to the osteodystrophy. So in hyperparathyroidic patient, what happened? Due to the continuous actions of the parathyroid hormones, there is a continuous release of calcium from a bone, so which lead to the osteodystrophy. So high calcium level affect a neuromuscular excitability, so which causes a fatigue, a mental disorder, depression, and heart dysfunction with a certain cardiac QT interval in the electrocardiogram. So low level of parathyroid hormones, that is in a hypoparathyroidism, so what happen? There will be the convulsions or high activity, and when there is a this high level of calcium is present, that it downs the acceptability. So that means it lead to the depressions and certain cardiac QT intervals. So this is all about the this parathyroid hormones.